Hello, everyone, and welcome to today's webinar. This is Megan Weber. I'm an extension educator focusing on aquatic invasive species at the University of Minnesota, and I will be your moderator today. I am looking forward to having Ranjan Muthukrishnan join us today for our webinar. Ranjan comes to us from Boston University. He's a research assistant professor there, and today he'll be sharing his presentation, Understanding Starry Stonewort Invasions in a Changing Climate. Welcome, Ranjan.
I'm excited to get to tell you a little bit about my work on starry stonewort, and particularly how this invasion might be influenced or interacting with climate change. It's work I've been doing for the past number of years, largely in coordination with MASERC, and I really appreciated their support. And we've got really new data that's just hot off the presses, and so it's kind of fun to get to share it with you all. And I just want to start off by taking a second to thank all my collaborators and partners on this project who have been really important in introducing me to the system, like Dan, but also developing this project. And then particularly Carolyn Kalinowski, my technician, who really ran most of the fieldwork of this project. And without her, 100% this project would not have happened. Okay, so just a little bit of an overview and outline. What we'll talk about today is I'm going to give you a little background about starry stonewort, a little bit of the background rationale of the project, our approach, and then our initial results from what we've kind of just done the last few years. And I imagine many of you probably already know a bit about starry stonewort if you've taken the time to come to this talk, so I won't belabor it too much. But it's also known as Nidalopsis obtusa or N. obtusa. I'll probably interchangeably use those terms, and you'll see both on slides at different places. It is a green macroalga, which is sort of the algae or kind of the more ancient lineage to most of the flowering plants that you see, although certain times I'll probably talk about it as a plant itself. It's a charophyte, or it's in the group Cariaceae, which is closely related to the stoneworts and muskgrasses, which are native to Minnesota. You might hear a lot about the Caras, another really closely related group. And these species are ecologically important. They can really influence water quality. They can also provide habitat for other organisms, and particularly fish. So these native species are really important, and so interactions with them are things that we might care about. A little bit of the invasion history of starry stonewort. It's a relatively new invader to both North America since the 70s, which is not, I guess, that new. Originally in sort of New York and more on the East Coast. It's been moving its way westward, and it first arrived in Minnesota in about 2015. Or I should say, that's when it was first identified. It likely was in Minnesota, I would bet, well before that. It's quickly gaining ground. Like I said, it's been moving across states quite effectively. Every year we seem to find new locations in Minnesota, and it's becoming somewhere between increasing and significant concern for AIS management. And it's really, our expectation is that it's largely transported by boaters. It can really easily get caught up on props of a boat, and you can move it from one location to another. And if you see this sort of white star-shaped picture up in the top, that's what's referred to as a bulbil. It's not actually a seed, but it's kind of a reproductive propagule that it can sort of, that bulbal structure can create a new individual, and either in the same lake or another lake. And so if that plus some of the plant material gets caught on a prop, it's really easy for it to move from one lake to another. And this is our first moment for totally fresh. You guys are the first ones to see it, data or results. That's really fun. So, mostly we think of it as being transported by boaters. We also tend to assume starry stonewort doesn't get eaten by many things, but I can show you some of our cool first initial results of these, what I'm pretty confident are duck poops, that I also believe are just chock full of starry stonewort. This is from a bed in Crooked Lake in Indiana, and there were just a whole bunch of these little pellets sitting on the top of the starry stonewort beds, and you can see they're just full. It's just a total mass of starry stonewort. I think I've got a little video. You can see one sort of coming apart a little bit. So I think it's pretty clear that something, and I'm guessing a duck or a goose or something, was eating them and pooping them out. And theoretically, if that bird moved from one lake to another, that could be both a mechanism of dispersal, and also if something's eating it, it could be acting as a food resource for members of the food web. So that's just a fun little thing that we saw. Anyways, so the overall distribution of starry stonewort, you can see, again, started in sort of the New York region along the St. Lawrence River, and then it's been moving its way west, I think in Pennsylvania, Michigan, Indiana, Wisconsin, and now Minnesota. And it's probably even been identified in a couple more places since this particular map was created. And if we look at Minnesota specifically, I think I've updated this to the current distribution of 19. Now I can't just say lakes, it's water bodies, because it's also been identified in the Mississippi River, I believe near Wolf Lake. And a number of these lakes that have been where starry stonewort's been identified have actually been identified through Starry Trek, which is also put on by MASERC. So they're doing a great job. And I just take a moment here to comment that, most of you can see these sort of two clusters, and most of that northern cluster of lakes are actually on tribal reservation land, particularly I think Leech Lake and Upper Red or Red Lake. And so in fact, all of these locations are in, historical tribal lands. So it's really important to me and to our group that we do this type of work in a way that is both useful to and honoring of those communities. And so we want to always be aware and thinking about those issues as we do this work, especially because a lot of it happens on tribal land or on reservation land. Anyway, so when I think about starry stonewort and why one ought to care about it, there are a couple issues that are important impacts related to starry stonewort. And the central ones, or really most of the impacts, they come from this idea or this situation where starry stonewort can just grow in really, really dense aggregations, really large abundances. And in those really dense aggregations, it just sort of chokes off all other, there's, very few other plants can be mixed in there. It's so dense that it's hard for, boats to go through it. It's hard, you don't really want to swim around in it. In some cases, you can see, especially in that lower image, it can really grow all the way up to the surface. Some years it's well down below the surface, but in some cases it gets all the way up to the top. And so it can really just influence or impact negatively recreation, be it swimming or fishing or boating. It's just kind of gunky. Nobody really wants to be up in all of it. And then the other set of impacts that I really think about are what it does to native communities. Like I said, it can grow in these sort of really dense aggregations that lead to a loss of diversity. You sort of displace that sort of diverse native community with this much more low diversity, if not monospecific, starry stonewort dominated community. And when you have that loss in diversity, we know from other studies is that the systems tend to become less resilient, and they provide reduced services, if that might be in water quality management, certainly in things like fishing, and those sort of recreational services, but also sort of more ecological services as well. And so all those are generally negative impacts that we would like to prevent. So to sort of get into this question, or to sort of get into the both effects and sort of dynamics of these invasions, a handful of years ago, I started out by putting together a pilot study to look at spread and expansion of starry stonewort, initially in Lake Coronis in sort of central Minnesota around St. Cloud, Alexandria. And the way that the project works is we found areas where you had a starry stonewort patch kind of up against or adjacent to a bunch of native plants, and so where you'd see this is kind of what's diagrammed out there in that little blobby image on the right side of your screen. We looked for places where there's starry stonewort near the native species, and then we would put in physical posts to create a 30-meter transect that we could then essentially run a transect tape, or essentially a long measuring line between those two posts, and then we could go along it with this essentially square quadrat, and at every meter we could look and see what plants are there. And let's see, the idea was to be able to monitor sort of the change along that transect over time. And this way you can see initially when we did in Lake Coronis, we put up 5 separate transects. We measured and what the color here is essentially saying how much starry stonewort there was. And the idea was they always started in an area that was starry stonewort dominated. They transitioned into an area that was native dominated. And ideally they would have sort of a sharp transition point, which is right at the edge of the invasion front. And then the idea was over time, we would sort of watch how that that invasion front progressed, and we could measure its expansion rate. And so some of you may have seen this next bit of data that was presented a couple of years ago at actually one of the other Macerc showcases by Carly Wagner, where she showed that between 2017 and 2019, there was this really dramatic expansion of starry stonewort, basically completely taking over almost all of the transects, just in a single year, it just completely taking over. I wasn't actually there, but I heard that there was like an audible gasp in the room when the data was shown, and everybody just was really surprised, if not, and it was really stressful results to see that, like, thinking that starry stonewort was just going to expand and take over so rapidly and dramatically. And I think reasonably so, that like that was such an exceptional spread rate and expansion that it felt like this could be incredibly problematic. And in particular, it sort of aligned with our understanding, this general understanding of how invasions work, that you start off, a location that doesn't have any invasive species, and then you get an establishment event, and you have pretty rare, very localized amounts of starry stonewort. And it has a bit of a lag phase, but then it starts to expand really rapidly, and it will just take over and become widespread and abundant. And if that's the case, then as soon as you get your invasion, you probably don't even know it's there until it started increasing, and at that point, the worry is that it's sort of too late. But I really wondered, particularly in these aquatic systems, like, is it always this bad? We definitely saw, again, this is on Lake Coronis, situations where there were these super abundant, large, almost floating pillows of starry stonewort all the way to the surface, really gunky and just really problematic. But the question is it always this bad? You have some variation across a lake, and in fact, I'd been on Coronis in a couple of different years, and it didn't always seem to be quite this quite this bad. And so I was interested in the question of, does or can biomass go down? I realize I've made a slight change in between these two slides. Can biomass actually go down in certain years? Or is it always just going to be really high once it establishes? And then secondly, if there is some variability from one year to the next, does the climate matter? Are those conditions important to whether or not you have a lot of starry stonewort, or are there scenarios when you might have less? So the approach that we wanted to take I'll go over both some of the hypotheses we have and then some of the approaches and activities that we did. So our first hypothesis about climate and starry stonewort is that climate would actually influence the phenology, which is sort of just a fancy word for the seasonal patterns of growth and when does it start growing, when does it hit its peak biomass, and then how quickly does the biomass die back at the end of the season, and when does that happen. And we thought that under different climate conditions those patterns might be different. We also had this hypothesis that in warmer years, with climate change, that could actually increase the biomass and spread of starry stonewort, with the idea particularly that really long growing seasons can support just extended expansion or growth of starry stonewort, potentially making it worse. And then the idea being that increased starry stonewort would then end up displacing native plants or native species. So what did we do to sort of look at this? First, we wanted to establish transects just like we did on Lake Coronis, but we wanted to use a broader latitudinal gradient to try and think about what happens under sort of warmer, cooler climates. So we went all the way from northern Indiana all the way up to northern Minnesota. It's about, I want to say about 1,000 kilometers is that distance, like north to south. And then we put in those same types of transects where we could then measure plant cover and starry stonewort expansion, just like what I showed you a moment ago. And then the other thing we wanted to do was actually take biomass samples so we can measure, again, those seasonal patterns, as well as year-to-year differences in the overall amount of starry stonewort. Again, another way of measuring, are there good years and bad years for starry stonewort, as well as native plants. So again, this is that Lake Coronis data that we saw. And the initial results we got from 2020 showed actually that you did have some reduction in locations. It wasn't this constant expansion and persistence of starry stonewort. At least in a couple of different locations, we actually had reductions. So you go again from that dark red, which is just 100% starry stonewort, to a couple areas that are blue or at least kind of yellow and sort of mixed, sort of intermediate amounts of starry stonewort. So that's Lake Coronis, but we wanted to do this not just there, but across our whole set of lakes. And so this is our new experiment that we set up in 2019. You can see there's Indiana, Wisconsin, and Minnesota lakes. I will note that Coronis actually isn't included here because we weren't able to do all of the sampling in 2019 there. And so we couldn't do all the analyses. So that, and it's also a bit of an outlier. So it's not included on this data set. But so that's 2019. And then you can see we went back in 2020 and 2021. And I get that these are very messy graphs. I'll help walk through them in a second. But the main thing that I'll point out is in 2019, there were, again, sort of that nice, more or less those nice invasion fronts that we were kind of shooting at, where you had starry stonewort on one side and native species on the other. But then in these subsequent years, it gets kind of quite a bit messier, which was not entirely what we expected. And what you can actually see is there's a lot of places, particularly in 2021, where there was much less starry stonewort than there was even initially, which again kind of goes against our initial instincts. But again, this is a really complicated, messy thing. It's hard to sort of understand exactly what the changes were. So what I did is essentially I asked at every individual location across every single one of those transects, did starry stonewort go up or down? And that's what these two graphs are. And one is the 2019 to 2020 change, and then 2020 to 2021. And what you can see here is if you have a spot that's green, that basically meant that there was no real change in the amount of starry stonewort. Parts that are orange, yellow, and red meant that you get more starry stonewort, and then the areas that are blue are actually losses in starry stonewort. And so again, like we actually have quite a few places where there are some changes, but it's interesting that it goes kind of both ways. Sometimes you're getting expansion, sometimes you're getting reduction. Sometimes, like on Crooked Lake in 2019, you have both of those things happening on in the same transects. Some parts of them are expanding and some parts are reducing. Um, but you have a lot of variability, a lot of change. And in particular, I want to point out in 2021, we actually had some pretty significant reductions in particularly in those three lakes, Pike, Winnibigoshish, and Moose, which are also our most northerly lakes in the study. And so it might suggest something about those more northern lakes being a little bit more resilient, being able to I don't want to go quite so far as to say fight off starry stonewort, but maybe those are places where starry stonewort can't persist quite as easily. So yeah, so that's our first set of new results that we got. But we want to sort of interrogate, these are the overall changes. Can we try to understand a little bit more about the dynamics and why some of this might be happening? So first, the first thing I wanted to look at was this seasonal phenology, the sort of seasonal patterning of growth. And we had this expectation that starry stonewort, it's sort of been known to have this distinct late season phenology compared to other both native species and other invasive species. Like you might know, curly-leaf pondweed is actually known for being a really early season growth. Like it expands in like April, May, but then is pretty much died off by the middle of the summer. But we'd had this expectation that starry stonewort really works, grows much later in the season. And so we wanted to see if that holds across different years. And we were also benefited because we did have some prior and historical data that we could bring to bear on this as well. And so what I have here is actually, we were able to get some data, again, from a bunch of different years, including one from the 1980s from a study by Nichols et al. And I won't say that this is pre-climate change or anything, but it was quite some time ago. And you could think of it as a little bit closer to a baseline of what, we might expect the annual patterns to look like. And you can see it has this pretty late sort of peak in biomass around September 1. And then that biomass actually stays pretty high all the way through to the end of the calendar year. And then in 2017, from Lake Coronis, we had some data from that paper I just showed you. And again, it starts growing maybe a little bit sooner, but it has this, again, this peak that sort of, it plateaus a little bit. It sort of stays high for quite some time, for a few months. And it's again about at the same time, maybe a little bit earlier than the 80s data. And then in 2017 and 2018, from again Coronis and Moose Lake in northern Minnesota, we see again that kind of same later peak, but you can see that it rises rapidly and then falls rapidly. It doesn't sort of keep that longer plateau for multiple months. And then in 2021, and I should say, we kind of had a gradient with 1984 being relatively cool, 2017 being warmer, 2018 being a little warmer than that, and 2021, as you might know, was sort of an exceptionally warm year. And so when we looked at the data from this year, you can see, again, this is from all of the lakes that we sampled this year, 9 of them. And the key things you can look at is actually that you had this much earlier shifted peak. Our peak biomass was kind of in early to mid-July rather than our sort of late August sort of expectation or early September expectation. And then this year, as well as in 2018, we had these really rapid diebacks after you hit peak biomass rather than these sort of longer plateaus of high starry stonewort biomass. So that was our first thing that we saw, this very, I think, pretty clear shift in phenological patterns, like pulling earlier in the year. But then the other question that is really important and interesting to us is just the overall biomass. How does that correlate with temperature? And this was actually a really surprising result that we had. And so we've kind of aggregated data from all of our different lakes and multiple different years. And the colors here tend to identify individual lakes. And what you can see in this kind of surprising result is that in hotter years, we actually tended to have lakes have less starry stonewort. And this includes both, from individual lakes that get measured from one year to the next. You can see that the warmer years tended to have less, but then also our more northerly lakes or our more southerly lakes, which are also generally warmer, also seem to have lower overall biomass values. And so this is not at all what we expected, although there could be a handful of reasons where you might actually imagine that warm conditions are bad for starry stonewort, or might be marginally better for native species. And I'll talk about some of those in a bit here. But I will say this is sort of a complicated sort of data set and a complicated pattern to think about, or to unpack like what's really driving it. And I will point out, if we look at all of the lakes individually, so there's sort of 3 years of data for each lake, they're often really influenced by a single really low abundance year, which was, this past year, 2021, which was not only really warm, but also a really dry year. There was a lot of drought conditions, which, go along with each other often. But the fact that, it was sort of such an outlier year, we have many lakes that the lake level was really low. And it's hard to know, was it a function of drought or temperature? And how might we, extrapolate out into the future based on those things? So just a little bit of caution from just assuming that you're going to have low starry stonewort whenever the temperatures are high. But that is sort of the pattern that we're tending to see in terms of overall biomass. But biomass is just one thing that we're interested in. By looking at those transects, we're also interested in the sort of overall cover of starry stonewort, because that's actually a really strong, metric of the competition between starry stonewort and native species. And in a lot of ways, like the health of those native species are what we care about as well. And so if we look at those transects, what we have here is, this isn't overall biomass, but it's that from one year to the next is the coverage, of those transects, like what percentage of those transects have starry stonewort on them versus not. And here, when you look at how temperature correlates with year-on-year changes in coverage, we actually get some sort of conflicting or inconsistent results. Certainly in the summertime, much like for biomass, when you have high summer biomass, when you have high summertime temperatures, that tends to be reductions in starry stonewort coverage. But at the same time, if you look at the winter temperatures of the year prior, or the winter prior to summer, there actually seems to be a positive correlation that warmer years are actually beneficial to starry stonewort, which kind of was our initial hypothesis. But again, you've got this inconsistent pattern that, depending on whether you think about summertime temps versus wintertime temps, and there's reasons to imagine that those things might influence the system in different ways. And also, starry stonewort was just one part of the story that we want to think about. We're also, like I said, really interested in the native species. So if you look at that same analysis, but we look at the coverage of just all native species, we see effects that are kind of similar in direction, although maybe a little more different in magnitude. And here again, you see like warmer winters are actually really good for starry stonewort, and warmer winters are good for native species, and warmer summers are bad for those native species. The same is for starry stonewort. suggesting maybe it's not so much the competition between starry stonewort and those native species, but just they're all acting the same. It's just, is it a good year or a bad year for plants? And so then you really want to think about how does the competition between those two groups play out, rather than just, oh, is it good for or bad for either of them? And also, much like for starry stonewort, we looked at biomass. If you look at biomass for the native species, it actually has much weaker effects. Wintertime temperatures seem to have almost no correlation with native biomass, but you have a slight positive correlation for summertime temperatures. So again, like there's some complexity as to whether warmer years are good or bad, and how does it influence the competition. But it does at the same time seem pretty clear that there are some relationships, and likely they're complex because they influence not just sort of the growth of an individual species, but of the whole community, and then of the sort of interactions between species. Okay, so that was a ton of data that I threw at you. We're sort of thinking through it ourselves. We just got some of this data back. And this is the point in the talk where I'm supposed to think about all that data and have some sort of synthetic opinion about what to make of all of it. And I'm not 100% sure. One, it's kind of new to us. We're still thinking it through. And two, it's really complicated data, and we have a somewhat limited number of locations, or we have a decent number of locations, but a somewhat limited number of years. This 2021 might have been sort of a particularly anomalous outlier of a year. So it's complicated. And there's reasons to imagine that it could go one of multiple ways, be it, the fact that starry stonewort grows really late in the season, maybe all the sort of warm, good growing conditions early in the year really benefit the native species, and that sort of allows them to have strong competition against starry stonewort. Maybe it has to do with, ice out dates, or maybe it's just so hot this year that that's crappy for everybody. So, whatever, there's some uncertainty, but there's reasons to imagine that this could go different ways. But I'll try to come up with a few overall conclusions. The first thing that I think is pretty clear is that there was a lot of variability in biomass, both between years and between lakes. And the expansion of starry stonewort is not constant every year. So those assumptions, those expectations we had about this constant one-directional growth of an invader really seems to not be holding true in this system. And it's interesting why that might be. But the other thing that I take away from that is, well, there's some potential for resilience. It's not just invaders get here and then they get worse. There's going to be some years that it probably won't be as bad. I'm not necessarily going to say that, like, oh, well, with climate change, like starry stonewort is going to stop being a problem. But it might be useful to think that there'll be better years and worse years. And so maybe if you were thinking about treatments, you don't have to do them every single year. It also might mean that maybe you wait for a low abundance year, and that's an optimal time to do a treatment because you can get the most bang for your buck. I don't know if that's necessarily true, but maybe, but I think that's a question that sort of is still open and ought to be explored. We did certainly see these earlier peaks and faster declines in warm years. It might, again, be this idea that starry stonewort kind of hits its max, and then eventually it just, in these warm years, it just can't sort of hold on. But it does seem like there's a clear shift in phenology with these climate conditions. Similarly, it does seem like particularly warm years that there are these lower biomasses. But I think it remains to be seen if that's a purely starry stonewort thing, or if they're just tracking with the overall plant community. So those are the things that I feel fairly confident saying. But then a couple, sort of questions that still remain that we ought to be thinking about is, One is, well, how does climate influence this competition between starry stoneware and native species? It might be good or bad for either of them, but does it benefit or harm one or other of them more than the other? And then two, we just sort of, or I just in this presentation looked at or native species as a whole, but what about very specific native species? Are the Argyroceras going to act differently than the taller vascular plants? Are some that are going to do really well and some that are not? And also the other invasive species is Eurasian water milfoil or curly leaf. Are they going to be influenced in certain ways? That comes into play as well. Not as clear results as like we would have hoped, but I think some really interesting ones that deserve both some additional analysis and thought, as well as ideally, I think we could use with a few more years worth of data to try and to get a better sense of, were some of these results anomalous, or is that indicative of general patterns that we're going to see in the future? Okay, I just one more moment to thank my collaborators, partners, the people who help support us, MASERC, Indiana University, and thank you all for coming and spending a little bit of time listening to me. And if you have any questions, I'm here and happy to answer the ones that I have the ability to answer and give you best guesses on the other ones.
Wonderful. Thank you so much, Ranjan. That was fascinating. It looks like a really fun puzzle to keep noodling on. I'm going to take over the screen quick so that everyone can see the slide for our Q&A. If you have some questions for Ranjan, there have been many starting to come in. You can go to slido.com, enter code STARRY21, and that will get you in. You can upvote a question that you find interesting, add your own in. If you have a phone or other smart device, you can scan that little QR code and you can just do that straight from your phone too. And I'm just going to go ahead and get started with the questions. What I think I'm going to start with, because there were a couple along these lines, is there's a lot of interest in the duck poop. So I think I'm going to try and lump the duck poop questions together. So the questions are if there were bulbils in there, and if there was any viability tests, and the question on the viability test both related to if there's bulbils present, but there's also one about just the matter that was in there. And finally, if their dispersal would be possible unless there was like an intact node or bulbil in there. So three duck poop questions in one.
Yeah, so I will say that I did not actually see any bulbils in there. It wasn't a exhaustive search. It was me just sort of swimming around, probably while Carolyn was doing more useful work and I just was looking at stuff. And I just saw all these little weird things. And so I swam in there and took a look, tried to evaluate a little more closely. And so, so yeah, like I didn't necessarily see bubbles. That's not to say that there weren't. I do imagine that there's probably, it's less, well, if I was a duck and I was eating some plants, I'm probably eating at the top. And from pulling up a whole bunch of starry myself, I know that the bulbils are usually like well down in the sediments, and it would take a little bit of work to sort of really pull them up and grab them. But that's not to say that you couldn't, that a duck couldn't or wouldn't do it. And again, like with invasive species, generally, you don't need it to happen a bunch of times. It just needs to happen, one or a couple times. And there's like, I think the running assumption amongst our group is that a bulbil probably wouldn't be totally viable after being eaten by a duck. We always sort of refer to them, because they're sort of starchy, we kind of refer to them as sort of like a potato. And so you'd imagine that it might get, dissolved, digested in like a duck's digestive tract. But there was a lot of plant material in those little poops, and so I don't know how complete they're digesting them. Um, so I don't have any evidence that there was bulbils or that they would be viable, but it doesn't seem 100% out of the realm of possible to me. Um, okay, was there more? Did I catch everything about the duck poop, or was there another question?
Yes, I think that touched on all of them. If there's any lingering poop questions, I'm sure they'll end up in there, and we'll see if we get to them or not. But I will move on to the next question, which was wondering when you were showing the transects of the lakes in Indiana, Wisconsin, Minnesota, from that 2019 to 21 study, were those all from unmanaged lakes or unmanaged portions of lakes?
Yeah, so most of the lakes, but not necessarily all of them, I think probably have some level of management. There's a couple, like Pike Lake in Minnesota or in Wisconsin, I think they made a decision to not manage. Um, and I don't know some of like yeah, so many of the lakes have management, but maybe not all of them. But we had tried to set up our transects in places that we think are generally unlikely to get managed. We tried to keep them generally away from houses and stuff like that. And we talked with the DNR in most of these cases to try and either make sure that we weren't in areas or try to um, ask them to sort of prevent management in those areas. Um, and so I feel confident in most lakes that there hasn't been that our specific transects are generally unmanaged. There's maybe one or two transects in a couple lakes that might not be true. I think that but I'll say I don't think I don't think any of our transects get chemically managed, but I think there's some of the transects, particularly in Little Muskego in Wisconsin, that they have a pretty extensive mechanical harvesting program. And I think it's possible that the harvesters went over the plots. We don't know that that's the case, but it did sort of seem like that might have potentially happened one year. But mostly, I think that they are generally unmanaged, and I feel fairly confident that they're not chemically managed.
Great. Thank you. And I think that touched on a couple questions as well. So I tried to go through and clear out what all got answered in there. The next question is wondering about if you have plans for follow-up studies, if some of those are already in the works, and what questions from this one that have been raised are kind of your priorities for follow-up.
Yeah. So we totally have continued work planned. We have a grant. I will probably continue working on it in an hour as soon as this is done, to be submitted in about a week, to really keep a major part of this, well, to really keep all of this work going and then expand a bit more beyond that. So feel free to call your senators and have them tell the NSF to fund us. That would be awesome. But sort of the two, three major things that I think we'd want to keep thinking about and follow up are one, just I think that there's some value in like a few more additional years worth of data, just to get some more like year-to-year variability and changes to see. Again, like, if we have a broader range of climate or weather conditions, and just to see, if there were anomalous years or not. Maybe we'd get warm but not dry, so we'd still have higher water levels, to see if that was a driver as opposed to temperature. So I think just a little bit more data to get a little bit more of this climate variability would be important. The other thing that we really want to look into, and this would be in this follow-up study, is, as I just said, we kind of focus on unmanaged areas to try and understand the sort of natural dynamics of starry stonewort. But we really care about how effective these different treatments are. And so we have it planned that we would try and use these sorts of methods to get really fine scale data about the planet community, but in areas that management did happen. And we, purposefully go select those areas. One thing that I think that it's a surprising lack of data is, there's been a lot of treatments that have been happening. And in many of them, if not most of them, there is like some effort to, after the treatment to go back in and to look and measure and see, okay, well, what happened? Did we have a reduction in starry stonewort? But it's usually pretty soon after the treatment happens, like within that same year. And it usually has a pretty coarse resolution. They're usually these thrown rakes from a boat as opposed to like in the water surveys. Um, and and I think it'd be good to get a finer scale resolution of that data, but also, it's really rare that there's data that comes from a year later or 2 years later or 5 years after a treatment. With the exception of sometimes like treatments keep happening year after year and they keep doing sort of post-treatment surveys. But I want to know, like, if you do a treatment and you left it alone and came back 3 years later, in that scenario, did it work? Like, did you actually get not just less starry stonewort, 6 weeks later, but what about the next year and 3 years down the road? Or did starry stonewort just come back? Or, and then you can ask these questions about the native communities. Like if you had a thriving native community, and then you got rid of the starry stonewort, would they recover? Or could a treatment kill off the native plants as well, allowing starry stonewort to actually expand even more quickly? So yeah, these sort of longer term post-treatment measures is what I think would be really interesting. And then I guess the other thing that, plug is like, yeah, like this, I talked about half of this project, which is the ecological study, but it's actually all paired with social science work where we are interested in how people are making decisions about management. And we want to really dig into the decision making and sort of values components of the effects of starry stonewort. And so that's also embedded in these follow-up studies.
That sounds great. It sounds like we have lots of future opportunities for you to join us on a webinar again.
Yeah, definitely.
Let's see. So the next question was wondering about that 2017 to 2019 timeframe where you're talking about Lake Coronis and if there were management strategies that were happening on the lake at that time as well.
Yeah, so they did do I don't know, I don't remember the exact dates of which treatments, but they, so Dan Larkin, Wes Glisson, me, and some other folks, we had a paper that Wes led, actually looking at, I want to say they had 3 or 4 different treatment scenarios that were all happening on Lake Coronis, where they do mechanical harvesting. They had chemical treatments. I think they had chemical treatments with slightly different timings, like repeat or a single dosage, and then mixes of both those things. So they, particularly out of Lake Coronis, they really tried to do it in a scientific way to sort of compare these different treatments. And then we were able to actually go in and do, again, it was sort of same season post-treatment surveys, but and they have continued to do. I know that they have mechanical harvesters out there, or they did for many years. I haven't personally seen them recently. That doesn't mean they're not there. And so, but I feel confident that they've continued to do treatments, and I'm pretty sure they've done some additional chemical treatments. But again, like most of our sites were outside of those treatment areas. But they're definitely doing a lot of stuff on Lake Coronis.
Great. Thank you. Okay, next question was, wondering if you could compare the lake temperature data that you saw in your studies with what the temperature on the lakes in its native range in Europe?
That is a really interesting question, and I have no idea. But I am totally committed to finding out, honestly, um, because that's such a yeah, that's a really great question. Whoever the anonymous person was that said it, thank you. That's a great idea. I'm going to go look into it. The only thing I would say is, it's like roughly the same latitudes, and we have such a gradient of latitude and temperature within this data set that I bet it's somewhere in that mix, but that's just me kind of guessing. I honestly don't know, but I'm definitely going to look into it.
Great. Okay, and would you be able to share some of your thoughts, or if you at least have ideas on what the risks of starry stonewort invasions to wild rice might be?
Yeah, I mean, and this is a really important question to me. I was able this summer to go up to Leech Lake where starry stonewort has been found, and it's been found right mixed in with these wild rice beds. And I think that the risk is big, honestly. I don't know if that's going to be the case. It's quite possible that wild rice, being an emergent species that can get up above the starry stonewort and still get light, I think it has a better chance of being able to not get displaced by starry stonewort. We often see lilies that have a bunch of starry stonewort underneath them, but then the lilies more or less seem to persist. And similar to that, it was interesting, when we were boating around these wild rice beds, canoeing around, I should say, and I was really struck by how diverse and complex the aquatic plant community underneath the wild rice was, like sort of the submerged community in the beds was. And that's what was sort of getting displaced. When you have starry stonewort, you would lose all of that other diversity, but you'd still have a lot of wild rice. And so maybe it's going to be similar to the lily situation where you lose all the understory diversity, but the emergent plant, wild rice or lilies are able to persist. And I think that there's some likelihood that that might be the case, but it does make me wonder, what are the other broader sort of ecosystem influences? Do you lose resilience in some other way? Do you provide less quality habitat for fish, and then you have changes in nutrient cycling because you don't have fish? And also you probably have changes in water movement, which could influence nutrient dynamics. We've also seen, I wasn't able to show this data, but we took sediment cores in our starry dominated and native dominated habitats, and we haven't been able to actually process them in the lab, but like just literally looking at the sediments, you can see that they're way more organic. They are less, they're way more organic. They're much finer. They're less sandy in the starry stonewort dominated areas. And so there is, I think, some clear influence on the sort of physical processes and physical properties of the lake and the lake floor. And all of those could feed into the effects on the emergent plants. And so all of those things could influence wild rice. I don't know if they will, like that. I think we need to just do some additional studies on that. In fact, speaking of like future work, we are in the process of trying to establish some similar monitoring transects monitoring plots in Leech Lake to think about this exact issue.
Great. Okay, I am going to attempt to do another round of question lumping here,
Yes.
where we have a couple questions about water quality data and if you were measuring that as well. Two specific parameters that were called out were oxygen, and one wondering about if you noted calcium levels where you saw, in particular, declines in biomass, but also just questions about if you other general water quality data.
So unfortunately, we didn't take water quality data just out of time and resources. That's also actually in the larger proposal that we have. We're going to bring a biogeochemist on board to look at those things. The two things that I would say are, I don't know if, I think it's more, so I don't know if it's chicken or egg. I think that the dissolved oxygen levels will probably decline as a result of the presence of starry stonewort because it gets just so dense, and I think you get a lot less water movement. Um, but I don't know if like a higher or lower oxygen lake would be better or worse for starry stonewort. And also the fact that like it does grow in those super dense aggregations makes me think it could probably tolerate lower DO levels. Those are just instincts of mine. Um, and then the calcium, I don't remember, but I know that from another project that we did, we had a bunch of sort of water quality variables that we sort of were trying to correlate with sort of suitability for starry stonewort. And I don't think that calcium specifically really popped as like super critical, but I do think that there's kind of like a hardness, which is highly related. I think that there's a hardness um, effect, but I don't think it was one of the really strong ones, and I honestly can't remember it off the top of my head. But there's a paper in Aquatic Botany in like 2020 or 2018, something like that. That's the place to look.
Great. Yeah, we can try and find a link to that and get it in chat, or at the very least, send that out in a follow-up note. So I am just taking note of the time and that we have 1 minute left before closing. So I'm going to take that minute to do some of our closing housekeeping. First, of course, thank you, Ranjan, for joining us. This has been wonderful. There are many questions. People are clearly enjoying what you had to say. For those who have joined us, we did record this session, so you can watch for that. It will come up on our YouTube channel. If you want to subscribe to get notified, you can go to z.umn.edu/AISTube, and that's our channel. I will also be sure that a note gets sent out to everyone who registered when that recording is ready. Give us a little bit of time because we make sure to close caption that, so give or take, a couple days, around a week for that turnaround. Ranjan's email is showing. If you'd like to connect with him separately, I think he's up for that. It's there. And our program email address is also up. So if you have questions about the webinars in general, other programs and such, you can reach out to the team at aisdetectors@umn.edu. There are a number of questions that we weren't able to get to. Ranjan mentioned he might have time after that proposal he mentioned is in, to try and put in some typed responses to those. So we'll also send out messages if we're able to get some additional answers to questions that did not get answered. But with that, we're now 1 minute over. So I will say again, thanks to everyone for joining us. Thank you, Ranjan, for coming again, and have a great rest of your day.
Yeah, thank you for having me. It was great.